Hello, people. Can I see your faces real quick? How many people we got? 30 out of 50. Not so bad. Not so bad. Whenever I get more than half, I'm happy. How you doing, Bailey? I haven't seen you like laugh or smile in like ever.
Hey.
It's not like Australia, is it? It's work over here. Australia, it was all koalas and ski and surfboarding and
Yep.
time to knuckle down.
Yep. I just took an OChem exam this morning too. So
Oh, I feel so sorry. I think I aced every OChem I had, OChem test I had. I loved organic chemistry, and I love lording it over you people. Oh, I'm a jerk. I know it. I know it. You don't have to tell me. Sophia Martin, we haven't seen you in a while. DeJovany's not showing his face. That's okay. We don't want to see it. There's Sophia. Yay, Sophia. Looks like you got a man bun, except it's on a woman. What do you call that? You call it a woman. You call it a bun, basically, right? It's only a man bun.
A messy bun.
Messy bun. Okay. DeGiovanni, didn't you have a bun, or am I thinking about somebody else? No, that was Stefan McKean. Yeah, he's not in this class, I don't think. Casey Acevedo. Thaisa, come on, Thaisa. Let's see your face. You know, we all miss you when you're not here. Thaisa. Come on, Thaisa. Yes.
It's Taisha.
Thaisa in the house, yes. Hunter Reynolds. Now there's a quiet person. I've never heard Hunter Reynolds say a word. Let's hear you say a couple words, Hunter.
Hey, how you doing?
That's enough. Reminds me of an old joke, you know, the only president of the United States from Vermont was Calvin Coolidge. He used to call him Silent Cal because he said so little. And the story is he was sitting next to he was at a dinner party, and this woman sitting next to him said, I bet my friend that I could get you to say more than 2 words. And he turned to her and said, you lose. All right. Well, Barbara laughed anyway. Half of my jokes are for Barbara anyway. All right. So, welcome to the last lecture of cell biology. Tuesday, I'll do a review for the 4th exam, even for the final if you have questions about that. And Thursday will be the 4th exam, and then the following week will be the final. And then the world can kiss my butt for the rest of the summer because I'll be done. All right, so let's rock on some cancer here. Cancer. So the first thing to talk about, and I've kind of alluded to this on a number of occasions, but what is it that causes a cell to become cancerous? And you know, there are more recent theories that are different from this, but basically the model we're going on is it's DNA damage. DNA damage causes changes in genes, and the genes then can become, you know, knocked out or total or more active. You know, whenever you change the gene, odds are pretty good it's going to be knocked out, but sometimes it becomes more active. And that's kind of the basis for evolution, but it's also the basis for cancer. So when they started looking for genes that cause cells to become cancerous, they started out by looking at viruses that they knew caused cancer. And the first one was chicken sarcoma virus. So it's just a virus that causes muscle cancer in chickens. And they looked inside the structure of it, and they said, "Oh, look, this is a gene that looks like a kinase. It looks like a protein that's important." And they looked for it in humans, and they found out, "Oh my God, all our cells have this gene in it. Oh my God, we've all got cancer." But it turns out that you've got a lot of genes in your genome that could help you get cancer. We call these proto-oncogenes. If it's a gene that causes cancer, it's called an oncogene. But proto-oncogenes are kind of pre-cancerous genes, which sounds kind of scary, but honestly, what they really are is genes that make proteins that cause cells to divide. And you need those, right? Especially in development and wound healing and all of that. Everything we've talked about in terms of growth factors, all the genes that make the proteins in growth factor pathways are proto-oncogenes. And why is that? Well, let's look at them. So just going down the whole pathway of growth factors, first of all, you've got the growth factor itself, and there's an oncogene called VCIS, which is basically unregulated production of PDGF. So if a cell does that, it releases PDGF and it responds to it. It's self-sufficient in its growth signals. So any growth factor that is overexpressed by a cell and released can cause a tumor to form, okay? And the receptors, this is more common. Receptors can be mutated in such a way to make them constitutively active. So one mutation would be to overexpress the receptor, and that's pretty common in breast cancers. ERB-B, I think, is a breast cancer one. So if you make too many receptors, say you've got 100 times more receptors than they should be, those receptors bump into each other and activate themselves. Remember, the way the mechanism works is you're supposed to have the hormone there that then causes them to dimerize. But if you've got a ton of them in the membrane, they're just active. Also, you could get mutations that cut off the extracellular domain. That makes them active, or mutations that just make them bind together without the growth factor. The point being, they become constitutively active. They will never shut off. And that turns that proto-oncogene that makes the regulated receptor into an oncogene. G proteins, this is really common too, KRAS. So RAS is the G protein, and normally, it gets turned on by exchanging GTP for GDP, and then it activates RAF, but then it gets deactivated by GAP, GTPase activating protein. But if that step can't happen, if it can't interact with GAP and it doesn't have any internal GTPase activity, then once it's activated, it's just active. And that's another conversion of an oncogene, a proto-oncogene into an oncogene. Let's see. Kinases, any of these kinases in the pathway, especially MAP kinase, that's a really important one that gets mutated a lot. This is one we'll talk about later, BCR-ABL. This is from a chromosomal rearrangement, and it causes chronic myelogic leukemia. We'll talk about it some more later. But basically, any of these kinases get activated and can't be inactivated, that's an oncogene. Transcription factors, usually these are just overexpressed. Something like MYC. MYC doesn't have to be phosphorylated to be active, right? As long as it's there, it's on. So if it's overexpressed, boom, oncogene. And then all the other things involved in regulating the cell cycle can be oncogenes. MDM2 breaks down P53, right? If you overexpress MDM2, P53 is going to be broken down even if there's DNA damage. So that's another way something could become an oncogene. BCL2 blocks apoptosis. So if the cell becomes cancerous and it should go into apoptosis, it could overexpress BCL2, and now it's not going to go into apoptosis anymore, and it just keeps surviving. Okay, so these are examples of genes when they can get converted into, they get mutated, they get converted from a proto-oncogene into an oncogene. And just to repeat, any protein in those pathways we talked about, from growth factor to receptor to kinases to transcription factor, anything in that pathway, if it's no longer regulated, becomes an oncogene. And not just the RAS pathway, but the PI3 kinase pathway, and all the other pathways, basically. Now, the other side of it too is one thing that causes cancer is converting proto-oncogenes and oncogenes. The other thing is to knock out tumor suppressors. So tumor suppressors are any proteins that are involved in blocking the cell cycle, stopping the cell from dividing. So here, APC. APC is part of that destruction complex that that activates GSK3 beta to break down beta catenin. Well, if APC is mutated, then GSK3 beta never works, so beta catenin always builds up, that cell becomes cancerous. So basically a tumor suppressor is anything that stops division, and when that gets knocked out, cancer. PTEN, if you remember, PI3 kinase phosphorylates PIP2 into PIP3. PTEN converts it back to PIP2. You knock out PTEN, then, again, you've got a cancer cell there. And here's SMADs, which are tend to be growth inhibitory and TGF beta pathway. These two can cause cell division, but usually in the adult, they're involved in cell inhibition of cell division. Here's RB. Okay, RB blocks cell division by binding up E2F. If you've got no RB, then E2F just is active all the time. P53. Now this is more of a, it's hard, it can go into either one of these. So this is gatekeeper, right? So gatekeepers are the proteins that stop cells from dividing. You lose one of those, the cell divides. P53, I think, could go in either side of these. So caretakers then are proteins that fix DNA damage or stop the cell from dividing when it's got DNA damage. So P53, when DNA damage happens, it causes the transcription of P21, and that blocks, and that's a tumor suppressor. All your CDK inhibitors are tumor suppressors. So P53 can be considered a tumor suppressor for that reason. A gatekeeper type thing. But down here, we're talking about proteins that, like with the P53 pathway, when you found DNA damage, it would put out P21 to block cell cycle, but then also cause transcription of these proteins to repair the DNA damage. So these are all DNA damage repair enzymes. So they're called caretakers because they're taking care of the DNA. BRCA1, BRCA2, everyone's probably heard of those. Those are commonly inherited mutated genes that are found in breast and ovarian cancer. So if you've got one mutated version of a BRCA2 and then you lose the other version of BRCA2, your chances of getting breast cancer are a lot better. A lot worse, I guess, a lot worse. More likely anyway. So these are all, again, DNA repair enzymes. And here's an example of a guy with a XPA deletion. So this is an enzyme that's good at taking out thymine dimers, which happens a lot with UV damage. So he's got to be all dressed up whenever he goes out because any UV damage can't be repaired, and he can get cancer very quickly. So, okay, so you haven't spent a lot of time on this, but understand this is really, really important stuff. What is a proto-oncogene? It's a gene for a protein that will be somewhere in the growth factor pathway that causes cells to divide. And then that gets converted to an oncogene by being mutated so it's no longer regulated. Okay, that's probably the key of this whole chapter right there, if you know that. And again, it can be anything in this pathway. Tumor suppressors are things that block cell division. Two different kinds of tumor suppressors. One are the kind that block cell cycle, and the other are the kind that are caretakers that repair DNA damage. Okay, now that you know all of that and you're experts at that, let's go back to find out how cells actually get damaged. Okay, so what causes DNA damage? Somebody's not muted. And we're getting a lot of noise here. Let me see if I can see who it is. Thaisa, I don't think you're muted. Somebody's not muted. Oh, it's me. I'm not muted. Oh, okay. You jerk, Dr. Slish. Shut up. Okay, shutting up. All right. So what causes DNA damage? I'm glad you asked. One thing is chemicals, okay? Carcinogens are chemicals that cause conversion of proto-oncogenes into oncogenes. Anyone still swim in the Hudson River below that GE plant? I'm guessing no. So PCBs are a really big one. Polychlorinated biphenyls, the tars in cigarette smoke, or really anything that smokes. See, smoke, when you see smoke, it's basically particles in the air, and those particles are made by incomplete combustion of organic materials. So if you take organic material and completely combust it, you get carbon dioxide and water, which is pretty much what you get with natural gas, right? Natural gas is just methane. When you burn it, you may get some carbon monoxide, but mostly you're getting carbon dioxide and water. But anything more complicated than methane, you're going to get partially PAH, basically partial combustion products, polyaromatic hydrocarbon. That's our PAH. So the thing about cigarette smoke is you're taking, a drag of this every minute or so for your whole day. All day long, you're exposed to these polyaromatic hydrocarbons. So, so it's really, really dangerous. And you can see here how when people stop smoking, the rate of, okay, so this is people stopping smoking. Delayed beyond that is the decrease in deaths due to smoking, So, they'll also say things like, any kind of smoke is carcinogenic. So, a nice barbecued steak, carcinogenic, okay? But you're eating one steak a year maybe, or five, let's say. So I'm not that worried about my steaks. But smoking constantly all day. Also, as long as it's legal now in New York State, let's talk about cannabis. So cannabis is also carcinogenic, but let's face facts here. With today's strains of cannabis, you're taking 2 or 3 hits and you're good for several hours, right? You're not smoking a pack an hour. So yeah, it's also carcinogenic, but the amount of exposure is less with that, in my opinion, in my humble. Now, when we talk about carcinogens, there's 2 different types of carcinogens. Before we go there. So way back in chapter 12, I was talking about one of the specialized functions of the smooth endoplasmic reticulum in the liver is this cytochrome P450 system. And the job of that cytochrome P450 system is to convert organic chemicals into something that your liver can get rid of. And it's a very generic process. It's not looking for, phosphoglucose to take that phosphate off. A very specific enzyme reaction. It's very general. So it makes mistakes sometimes. And this is an example of that. You can take something that is not carcinogenic and add it to cells, and you don't get too many carcinogenic particles out of it. But you take that same thing, you run it through the liver, and now suddenly it's very carcinogenic. So this is kind of like chemicals that are pre-carcinogens. Sometimes your liver will convert them into carcinogens. And that's why, testing has to include this. I think this started back in the 70s when people realized this could happen. Dog. Okay, so that's the liver conversion thing. Here's the other thing. You've got 2 different types of carcinogenic things. One is called a initiator, and the other is called a promoter. So initiator would be something that causes the DNA mutation. And then a promoter is something that then supports that growth. So, for example, like croton oil, which has got 4-vol esters in it that activate protein kinase C. Croton oil by itself is not very carcinogenic. DMBA, which is a DNA mutator, it's a carcinogen, doesn't really cause that many tumors either. But if you first treat with DMBA and then treat with croton oil, now you get tumors. And so the interesting thing about this is cigarettes have both of them in, both initiators and promoters. So it starts your cancer and it keeps your cancer going. Okay, what else can cause DNA damage? Ionizing radiation. So we're talking about UV, and with UV, it's mainly your skin, right? It doesn't really penetrate. Also, things like gamma rays. Gamma rays will penetrate. Cosmic rays, which you're constantly being bombarded with. And this cosmic rays are one of the reasons why it's going to be difficult to go to Mars if you're a human being, okay? It's less than the distance and the time and the technological achievement of taking a human to Mars. Once you get outside the atmosphere, you're being bombarded by cosmic radiation, and there's really no way to shield it out there. So if you're out, you go to Mars and back, and it takes you 10 years, you're probably going to come back with cancer. So ionizing radiation, ultraviolet, gamma rays, cosmic rays, X-rays, any really high energy photon is going to cause this. UV, like that kid we saw with the whole skating with the whole suit on, UV causes thymine dimers. So basically it activates a thymine, which then interacts with a thymine next to it. And now you've got a kink in the line. Now enzymes will come and take that kink out, and they could put the wrong ones in. You know, they may do a double-stranded break, take both of these out, and then put the wrong thing in. So you do get you do get mistakes this way. Okay, that's ionizing radiation. And then something that's probably less important for humans than for other species is viruses. So viruses can cause cancer. And again, you know, certain viruses like retroviruses jump into and out of our genome, right? They insert themselves into the genome, and then they replicate off that genome to make more viruses. So if you look at a virus, and you can kind of predict its evolution, or not predict, you can kind of see its evolutionary history, where here was a relatively benign virus, but a retrovirus. It stuck itself into the human genome right next to a proto-oncogene. And then when it comes back out, it takes that proto-oncogene with it. Now it's a virus with an oncogene attached to it. And now it becomes a cancer-causing virus. So, you know, in the evolution of viruses, this kind of thing happens. So there are some virally caused cancer. Probably the most common is HPV, herpes papillomavirus. So you can get, you know, just herpes on your lip, you know, this virus. Really dangerous though is this one that affects the cervix and the reproductive tract. And the really awful thing about this is men don't get the cancer; they just give the cancer. We're giving. Men are always giving. No, but you know, all of you, and I think I have, no, I haven't, but all of you should get the HPV vaccine, both men and women, because like I say, men get it and pass it to women, and women get the cancer. So that's kind of uncool. Okay, so that's one viral cancer. All right, so that is how cancers are caused, mostly by DNA mutations, sometimes by viruses. How do cancers develop then? One thing you should know about cancer is if you get one proto-oncogene converted into into an oncogene, it's probably not going to make that cell cancerous. At the very least, it's not going to be able to escape the immune system and make tumors and become metastatic and everything. One little mutation is not going to make that big a deal. So it's kind of a stepwise thing. One thing gets knocked out, then another thing gets knocked out, and then you knock out P53, and suddenly you've got a lot of mutations. And over time, the cancers get more and more aggressive. So here's kind of an experiment that shows that, where you take this mouse and you inject some melanoma cells into it, and you get a few metastases in the lung. Then you kill that mouse, you take the lung out, and you grind it, and you take the metastases out of that lung, and you inject it into another mouse, and this one gets even more. And you repeat this over and over again. And the more you do it, the more aggressive those metastases get from one progression to another. And why is that? Of these cells, one of them, you may have millions of cells here that are making different tumors. One or two of them will get more mutations that'll make them more aggressive, and those come to predominate in the next one. So now you've got more aggressive ones. And here, one or two of these cells get more mutations that make them more aggressive. So in the next passage, they predominate. It's kind of like this cellular Darwinism. Every progression gets more and more aggressive. So it's a stepwise process, making cells carcinogenic. And, you know, they can play the numbers game, right? You've got billions of cells probably in a tumor, and they're all mutating. Most of them are going to die because mutations are going to be cancer, or mutations are going to be hazardous to the health of the cell. But some of them will get more aggressive, and those will predominate then. So the mutation strategy really works. Here is another example. This is colon cancer. So, by the way, looking forward to my next colonoscopy in 2 years. Woohoo! But only for the Versed. Oh, Versed. Okay, so this is the way your colon maintains itself. You know, you're always sloughing off cells. So what happens is you've got these crypts that come down into the stroma here of the intestine. And at the bottom of the crypt, you have stem cells that divide. Every time the stem cell divides, one cell goes this way and one stays a stem cell. And then so cells keep getting pushed up this way, and they're still dividing up in here. And then once they get up to here, they stop dividing and they differentiate, they become adults, they come up into the intestine. Now they're exposed to the exterior environment. Eventually they'll die and they'll be replaced from below. So your colon's constantly replacing itself by these stem cells. So in colon cancers, typically you get one mutation, like knocking out APC. Let's see. If you remember APC, it's part of the destruction complex that breaks down beta-catenin, right? And without APC, with Wnt around, that blocks that, and then beta-catenin comes up. Now, if you don't have APC at all, without Wnt, you know, you can't activate GSK3 beta, so beta-catenin is always building up. So APC is a tumor suppressor. Without it, you get beta-catenin and the cells divide. So here's an example of the stepwise development of cancer. So here's your normal crypt. So in one of them, you lose APC. So these cells are dividing too much, and they're just kind of building up here at the bottom. And then you get a mutation in RAS. So now you've got an overactive RAS and an overactive beta-catenin. Now the cells are really dividing and they're filling up. Now at this point, they haven't crossed this barrier, right? This is our basal lamina. So they're just kind of filling up the crypt and forming polyps that stick up into the colon. Polyps are not going to kill you, okay? They're just kind of like warts sticking up into your colon. But then you get more mutations, like PI3 kinase is active now, or PTEN is inactive. BAX is knocked out, so you can't get apoptosis. SMADs and TGF-beta mutations, now they become even more aggressive, and they leak into and they cross that basal lamina, and now it's malignant and potentially metastatic. And with colon cancer, typically, remember, everything from your colon goes to your liver first. So cells being released from these Malignant tumors will go to your liver, which you need on a minute-to-minute basis. So when your liver gets gorped by cancer, that's it. That's usually how it kills you. So anyway, bottom line being step-by-step process. You knock out one gene and you get a little bit aggressive, another gene gets more aggressive, and eventually you get enough mutations that it becomes metastatic, and that's what will kill you. Okay, so specifically, what kinds of mutations happen in cells? A lot of the mutations that happen during S phase in mitosis. So actively dividing cells are more susceptible to becoming carcinogenic. So your skeletal muscle cells, they're not dividing. It's extremely unlikely you'll get a sarcoma unless it's caused by a virus or there are some sarcomas, but they're not common. Something like 90% of cancers are epithelial cell cancers because epithelial cells have got stem cells, right? And so those cells are dividing. Every time it divides, it goes through S phase. Every time it goes through S phase, something can go wrong. And there's tons of repair enzymes to fix stuff, but things get through, things get through. So what kinds of mutations can cause cancer? One is a point mutation. So a point mutation is when you just change one base. So one base changes, and typically you change one base, it's either not going to cause a change in amino acid, or it could destroy the protein, but it could cause a hyperactive protein. An example of that, like I said before, is RAS. So your normal RAS pathway is RAS gets activated and GAP turns it off. Gets activated, GAP turns it off. You get a mutation, any one of these mutations, glycine, or glutamate in the 12, 13, or 61 position of RAS, and now it doesn't interact with GAP anymore. So once it gets turned on, it stays on, and that's your point mutation causing the cancer. Gene amplification. So I said this before with the receptors. So again, during replication, you get this gene amplification effect. So some genes you can see have been duplicated, many times, and they're way overexpressed in the chromosome. Some of that is just not harmful, and some of it is harmful. So an example of that is, like I said before, here's your normal growth factor pathway where the growth factor binds to the receptor and activates it. If you've got too many receptors there, they can just activate themselves or just be overreact to growth factors that are in the extracellular medium. Let's see. So again, breast cancer, what is it? Can't remember the name of the gene now, but it's basically the epidermal growth factor receptor gets overexpressed in a lot of breast cancers. Okay, chromosomal translocation. So this is when you get swapping of one chunk of a chromosome to another chunk of a chromosome. So here it's kind of like, here's chromosome A, here's chromosome B, this one cuts here, this one cuts here, and they swap, okay? So that's a chromosomal translocation. And it doesn't happen when they're like in this mitosis, totally condensed version like this. This is just to show you how it happens. It happens again during S phase when everything's spread out, things flip over, and then they maintain like that. So here you've got this chunk of this chromosome put over here. So these genes now are in a whole different environment. Part of controlling gene transcription is controlling how condensed or open the DNA is. That's called heterochromatin versus euchromatin. Heterochromatin is when it's all condensed up. Euchromatin is when it's all expanded out. And it can only be transcribed from when it's expanded out. So if you take this chunk of genes here that should be in a heterochromatin situation, and you put them into a milieu where they're in euchromatin, now it's spread out. Now you got genes that should be turned off that are turned on. So An example of that is in Burkitt's lymphoma, where you get a translocation between 14 and 8. So MYC is a protein that is a transcription factor, and it alone can make cells divide. Okay, it's a delayed, no, it's an immediate early gene, so it can activate cell division. So you put that, evolutionarily, this has been put in a really quiet area of chromosome 8. So it's only going to be opened up when it's needed. Mostly it's going to be closed up. Here's chromosome 14, which has got a lot of immunoglobulin heavy chains on it, right? And you know, in lymphocytes, this is a very active region of a chromosome because they're making immunoglobulins. So you take this MYC out of this quiet milieu and put it right next to this active region on a different chromosome. Suddenly you're making tons of MYC when it should be silenced. So this happens in some lymphomas. So those cells are now dividing too much because of this chromosomal translocation. This is another example. So this is actually my stepmother-in-law has this mutation. You take a chunk of chromosome 22 and chromosome 9 and you swap them. So you get a new chromosome here, which is part 9 and part 22. So the danger with this one is that right at this crossover point, you end up making a whole new protein. So over here on 9, you've got ABL, which is a kinase, and over here you've got BCR. I'm not really sure what BCR does, but here's your BCR, okay? You break it here, and you put this chunk of BCR onto ABL, onto this end of ABL. Now you've got BCR-ABL, which is a new protein. And this is a kinase. So this ends up on, oh no, it's the BCR has got the kinase, I'm sorry. So this now becomes an unregulated kinase. This kinase used to be regulated by some of these things down here, but now it's unregulated. So this gets expressed in lymphocytes and it becomes chronic myelogic leukemia. Here's the new protein. And here's the kinase domain of it. So again, I've talked in the last class about new generation of chemotherapy. This is a classic one, which is a fantastic series of drugs. These drugs basically bind to this kinase domain and only this kinase domain. So they're not going to mess up everything. They're only going to block this one protein that is the problem. So, for example, my stepmother-in-law was diagnosed with this at least 10 years ago, and she's been on these drugs ever since, and she's fine. She'll be fine as long as the mutation doesn't, the chromosome doesn't mutate and make it, resistant to the drug. So as long as the drug is working, she'll be fine. So new generation of chemotherapy. So, but my point is, again, chromosomal translocation. It can put genes into active areas where they used to be in quiet areas, or make whole new proteins by making fusions between things from different chromosomes. Local DNA rearrangement. So we're not talking about from one chromosome to another, but in one chromosome, something flipping around. And that happens a lot too. So here we are, chromosome breaks here and here, flip and maybe just deleted and close back up, or flips over, inverts and reinserts. So again, the DNA is changing, and now you can be losing important tumor suppressors or making new proteins. Here's an example of making a new protein. So if you remember back to chapter 14, control of skeletal muscle contraction. This is this is the actin thin filament, and tropomyosin is going across those myosin binding sites and being controlled by the troponin complex. See, this is a review for the final. So this tropomyosin is a coiled coil, which is a pretty common structure in biology, right? It makes a more stiff protein. Here's the FGF receptor. It's just got this extracellular domain that binds FGF, transmembrane domain, kinase domain. So here on this chromosome, I don't know which one it is, you've got the tropomyosin near the NGF receptor. And for some reason, it's vulnerable and it will commonly come out, flip over, and go back in. So when you do this, you end up with a brand new protein, which has got tropomyosin at one end, and you lose the extracellular domain, and then you've got the transmembrane domain and the kinase domain. So now you've got basically a tyrosine kinase receptor that's always going to be active, because once you put it in the membrane, it finds its mate. They bind together through that tropomyosin coil, constitutively active receptor. Okay, and that's, again, chromosomal translocation, which is flipping around, or chromosomal rearrangement. Insertional mutagenesis. So this is something that, let's see, when I was finishing grad school in 91, this is something I wanted to go into, but I ended up not, went in a different direction. The whole idea here is a virus, oops, shoot. Where do we start? Oh, okay, here. A virus can come, a retrovirus comes into your cell, and then it uncoats. The first thing it makes is a reverse transcriptase and turns its RNA into DNA. Its DNA then inserts into the genome, and then starts making more viruses from it, and then those viruses can infect and make more viruses. That's how a retrovirus works. So what can happen with a retrovirus is when it inserts into the genome, it's going to be active, right? It's a virus and it's going to be replicating itself. It could insert right next to a proto-oncogene and convert that into an oncogene. So that cell then becomes carcinogenic. Okay, so getting back to what I was saying, back in the day, we thought that retroviruses would be a great way to do gene therapy. So you've got a bad gene, and you put the good gene into a retrovirus. You then infect the patient with it and replace the bad gene with the good gene. Problem is, you don't know where that retrovirus is going to insert in the genome, and you could cause cancer. There is some utilization of this mechanism, but typically it's not a retrovirus. It's a virus that will infect the cell, make the protein, but then not replicate itself. So it's not inserting in the genome. There are some therapies that are involved with viruses as vectors for new proteins. But to actually change your genome this way is not smart. Today we've got CRISPR, so we don't have to anyway, right? You can go in to the exact gene you want to get rid of, disrupt it, put your new gene in the exact same place. So CRISPR-Cas9, send me a gene. Bum, 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 bum. Okay, so that's it for how let's review that real quick. The ways that genes can be mutated to convert to oncogenes, proto-oncogenes to oncogenes. Point mutation, gene amplification, making excess protein, chromosomal translocation, chunk of one chromosome onto another, local DNA rearrangements in one chromosome, or insertional mutagenesis via viruses. Okay, so that's 1, 2, 3, 4, 5, 6 different ways. Okay. Okay. Bum, 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 bum. Okay, summary. When I say summary, you know I'm almost done. For me. I'm gonna miss you guys. No, I'm not. Okay. I don't have any notes about this. All right, so summary. In summation, I would like to say the hallmarks of cancer. So this is something I talked about in the last class. Here's what causes cancer: radiation, infectious diseases. You can inherit one bad gene and the other one gets mutated out. Chemicals, that makes oncogenes, which if you inactivate tumor suppressors, that gives you genetic instability, so you end up with more mutations, okay, making more oncogenes. So you're accumulating genetic changes, epigenetic changes that are making cells more and more carcinogenic. So what does it mean to be carcinogenic? Self-sufficiency in growth signals. Okay, so So the cell doesn't need a growth factor to divide. It's already got its own growth stimulatory stuff. Insensitivity to anti-growth signals. So like TGF-beta usually blocks cell division, and this is gone. Evasion of apoptosis. Again, if a cell is dividing too much, it will usually go into apoptosis, either because the immune system finds it or internally it shuts itself off. And so you block those BCL2 proteins and it can get around that. Limitless replicative potential. So telomerase, making sure that the cells are immortal. Sustained angiogenesis, in order to get bigger than 2 millimeters, you have to get new blood vessels in there. And tissue invasion and metastasis. Once that thing starts to cross barriers like the basal lamina, it's become invasive and it is a cancer. So those are the hallmarks of cancer. Okay. What time is it? 11:46. Okay. So any questions? I'm going to stop sharing. I think I've shared too much already. Any questions or comments here? Nobody's got anything for me? How many people would like a quiz? I would. Okay. So I'm going to show that quiz and stop this record. I'm going to stop this recording and then have the quiz show. So you can go ahead and do that quiz. Once it opens, you've got, no. Once it opens, you will. Once you open it, you've got 5 minutes, and I'm going to shut it down in about 10. So get there right away and do it. That way, I just want to reward people who come to class. I don't want people to just do the quiz and get the bonus points. So I'm doing that now. Ready? Bye, people.